I hope everybody's doing well today. Thank you for taking your time out of your Saturday to come listen to my presentation. So today I'll be talking about oxidative activation of a calmodulin kinase 2. So my name is Daniela Barata. I am a senior molecular and cellular biology major here at Hopkins, and I work under the Mark E. Anderson Lab at the School of Medicine. Okay, so moving on. So the CaMKII mechanism, I'm going to refer to it as CaMKII from now on, the kinase. It's a pretty long name. So this is a really complex kinase. It works in very many, sorry if there's some noise in the background, there's a lot of honking going on outside right now. So this kinase is activated dependent on calcium spikes. So the frequency of calcium spikes in cells activates this kinase, which is really powerful. It's one of the powerful calcium sensors within many of our cells, specifically in the heart and the brain. So it can become independent of this initial calcium activation via a second mechanism of autophosphorylation. So this is pretty complex. We'll get into it in the next few slides. But basically, this is a very powerful kinase, and it undergoes a molecular switch-like behavior that contributes to learning and synaptic plasticity and memory. Okay. So CaMKII is implicated in a lot of pathology. Specifically, we will focus on cardiac pathology. So in the terms of arrhythmic syndromes, ischemic diseases, in addition to cardiac hypertrophy and pressure overload, reactive oxygen species, in addition to calcium, activates calmodulin kinase, which then turns on various different inflammatory pathways, such as the P38 apoptotic pathway, along with histone deacetylase phosphorylation, activation of many different cytokines and inflammatory pathways that reprograms cells to remodel themselves into a more inflammatory phenotype. So there's three goals actually, for the research that we're doing that unfortunately was cut short due to COVID-19, as I am sure is the case for a lot of us students. So the first goal was to study the molecular mechanisms of this activation of calmodulin kinase. Then we would find the potential mechanisms by which oxidation specifically can change the activity of CaMKII to result in the diseases we just discussed. Lastly, for the first aim, we want to assess how effective glutamine is as a model for oxidized methionine. So the oxidation event occurs in 2-methionine residues in the kinase domain, which we'll talk about later. So in order to mimic this oxidation, mutating to glutamine has been spoken about in many papers about being a good mimicking for oxidation so that we don't have to use reactive oxygen species in the lab. So our hypotheses were that the oxidation event would change the mechanism by which CaMKII is autoinhibited to weaken this autoinhibitory conformation and result in increased activity and basically prolonged activity of CaMKII and prolonged inflammatory pathway activation. And then lastly, we were thinking, well, maybe since oxidation results in hyperactivity of CaMKII, maybe we can have compensatory mutations in this kinase domain pocket, the binding pocket for the regulatory domain that can restore the activity back down to wild type levels and maybe turn off some of those inflammatory pathways. So we're going to delve into these areas of study, starting with just looking at the structure of CaMKII. So this kinase is really complex. So you see here, there's 12 subunits. It's a very, very complex enzyme. These are just 3 of the conformations in which CaMKII is found within cells, depending on its isoform or its microenvironment. We see 3 main parts: the association domain, it's like a central hub, bound to a linker domain, which is then bound to a kinase domain, the light blue circles here. We will be focusing on this little light blue circle up here for the following slides. Again, sorry for the sound. It's pretty loud outside. So then we're going to delve into the subunit structure. Like I said, this little kinase domain here. So This is a pretty busy slide, but don't get intimidated. I'll explain what this means. So the autoinhibited form we see here, this is the helical domain. I'm going to then redirect your attention to part C. So this helical domain, the green-blue, is extended out here, and we show, it's really loud. I'm sorry again, you guys. We see this inhibitory helix. So what I talked about before, how there's an autoinhibited state, and this regulatory domain is bound into this kinase domain. So this that you see here is this blue thing right there. So the kinase domain you see here in the inhibited form. So calmodulin is a calcium binding protein that's found in basically every cell of our body. So it binds this, once you add calmodulin kinase, this previously helical structure then becomes relaxed, and this previously relaxed structure becomes helical. So you see this here in the green relaxed state. This is the activated state. Calmodulin is now bound here. It bound in this relaxed place to then structure it into a helix. Once calmodulin binds this, it relaxes and opens up this inhibitory regulatory domain. So then here in this little threonine residue that we see here, this little dot, you have a transphosphorylating activating kinase. So what that means is that, I'm going to go back to this slide here. This kinase can actually, once this kinase is bound to calmodulin and activated, this adjacent kinase goes ahead and phosphorylates this other kinase. So it kind of cooperatively phosphorylates this one, which then phosphorylates this one, etc. So this becomes an activated CaMKII. So it's a very complex mechanism, but that's pretty much all we need to know for the terms of this study. All right, so we're going to go into the sequence of this little regulatory subunit here. We're going to delve deeper into it. All right, so the R1 sequence is what this is called. So these are all different isoforms of CaMKII, just different versions of the same enzyme, which has very slight differences in the sequence of amino acids. So we're going to focus on these green residues here. So this is the calcium calmodulin binding region. And you see here an ox, which means oxidized, and it's the methionine at position 281 and 282 that gets oxidized by reactive oxygen species in the cell. So in the lab, we used CaMKII delta, which we see here. In the first row, and the sequence at 281, 282 is methionine. So cysteine here, CM, we see cysteine is also oxidizable, similarly to methionine. These are all just different isoforms. And you see here, there's a lot of ionizable residues near this 281, 282 position that suggests that this region is very responsive to the microenvironment of the cell. So lastly, we're going to direct our attention here to the kinase domain, which binds, you see this in yellow here, is actually the regulatory domain, the autoinhibited state of that regulatory domain. We see tyrosine, we see glutamine again. These are all ionizable residues that confer the binding of that regulatory domain to the kinase and allows for the inhibitory association. So we were thinking, since the oxidation of methionine creates a different environment here that makes it unfavorable for this autoinhibition to bind, this autoinhibitory binding to occur, maybe we could in the future mutate some of these ionizable residues to favor the reassociation of that oxidized state of the regulatory domain to then bring down the activity and kind of like, hey, it's oxidized, let's bring the activity down by mutating in a way that can reassociate that oxidized regulatory domain back into the kinase domain and kind of calm down the kinase. So lastly, we're gonna look at a model for oxidized CaMKII activity, which first we're gonna go into unoxidized CaMKII activation, just how it's normally activated. Again, we see here, this is calmodulin kinase binding the regulatory subunit. Usually prior to calmodulin binding, this relaxed structure here was bound back into its kinase domain in the autoinhibitory association that we have discussed prior. So again, revisiting that. Calmodulin binds that helix, then relaxes this section, And the threonine that we find here is then phosphorylated by the adjacent kinase. So just revisiting that model for activation. Once this is phosphorylated, the interesting thing is that calmodulin can actually leave. It can unbind, and the kinase remains active independent of calcium. So it's really, really powerful. Once it's phosphorylated and once it's also oxidized, this similar interaction occurs where calmodulin can then unbind and it remains active, and all of these inflammatory pathways continue to be active, which is really important. So yeah, basically that was an overview of how calmodulin kinase 2 is activated. Okay, so here, this is just a little bit of background with a prior study done at this lab where they hypothesized that the calcium calmodulin binding kind of opens up that regulatory domain to allow threonine to be accessed by the kinases. So they proved this by adding reactive oxygen species to CaMKII, but eliminating calcium calmodulin from that pretreatment, which shows that there's no enhanced activity of CaMKII. They now rescued that by then pre-treating the cell with calmodulin kinase and then adding reactive oxygen species, and they did see persistent activation. So this basically proves the association of calmodulin kinase exposes the methionine residues to allow for the oxidation event to occur. Okay, so this is just some more data to show the importance of oxidized CaMKII in pathogenesis. So again, the same lab that I work with published this paper in 2008 that showed angiotensin II as one of the oxidizing factors for CaMKII, and it led to apoptosis of cardiomyocytes, both in vitro and in vivo in mice models. Then it is important to note that methionine sulfoxide reductase is actually able to reverse the oxidation of CaMKII. So they knocked out this methionine sulfoxide reductase and showed that there was increased oxidation of CaMKII because this rescuing reversal could not occur, led to myocardial apoptosis, impaired cardiac function, and increased mortality after myocardial infarction. So basically, this is all just very overwhelming evidence that oxidation and reactive oxygen species leads to pathogenesis that is directly correlated to calmodulin kinase. All right, so the second aim of this research, which unfortunately was cut very short, was to characterize the difference in enzymatic properties between the oxidized and unoxidized CaMKII. So again, we mutated the CaMKII methionine residues to glutamine because it has been shown to be a good model to mimic oxidation. And we assessed the potential mechanisms by which kinase domain mutations could allow for restoration of activity. That hasn't been done yet because, again, COVID cut everything short, but we hope to do that in the future. So we created a spectrophotometric assay that tested for Vmax, cooperativity, and all of these different parameters for CaMKII activity. So this is just overview of the mechanism. There's no need to understand everything completely here, just to know that we used a lot of metabolic enzymes and metabolites to create a non-radiative spectrophotometric activity assay. So the main idea here is that we have NADH added to the reaction, and as CaMKII is active, it takes up NADH and reduces the absorbance of NADH, which is then going to become a measure of CaMKII activity. So basically, at 340 nanometers, we can measure the absorbance of NADH, and if we decrease absorbance, then we know CaMKII is using up these metabolites and working. Okay. So this is just the results from one of the initial CaMKII activity assays. And we have many different conditions. So the first was just wild type CaMKII, then just a smaller concentration of CaMKII, which correlated with less activity, which makes sense. And then again, a third smaller concentration. Now here we started to see, knock things out to see if the assay is working the way it should be. So if we don't add any CaMKII, we see basically no activity, which is consistent with what we expected. Then we did a no ATP condition, which you need ATP for all of these processes to occur in the assay. So again, no activity as expected. And then finally, we knocked out calmodulin kinase. Calmodulin and calcium, we knocked that out and did see about 25% autonomous activity, which we hypothesized was likely due to the autophosphorylation remaining in these kinases, since they work pretty independently and can remain activated. We said that maybe we should mutate this threonine to alanine so that it's not phosphorylatable. But that also creates a limitation because it might mask any oxidation and phosphorylation like interdependency for the activation. And then finally, Syntide, which is the substrate for CaMKII, we knocked that out and obviously no activity. So this kind of proved that this assay is a functional assay and could be useful to measure oxidized CaMKII activity. And then finally, last slide, we measured DNA actually as a CaMKII activator, because one of the effects of reactive oxygen species in cells and oxidative mechanism is that you find mitochondrial and nuclear DNA in the cytoplasm of cells, which if that's happening, your cell is in trouble. So we see CaMKII could be one of the DNA sensors in cells which responds with these inflammatory pathways. So we see here that in the minus DNA condition, you see slightly lower activation of CaMKII due to the decrease in absorbance of NADH. Oops. And then in the red, we added DNA and we see increased activity of CaMKII in almost basically every condition except for the 0 nanomolar calmodulin, which makes sense. So finally, we just want to give some sort of like future directions. We want to finalize this assay to make sure that we can characterize reactive oxygen species and DNA as activators of CaMKII and implicators of pathogenesis in the heart, and run a finalized assay on this glutamine and methionine CaMKII, and maybe use crude lysate from tissues such as mice heart tissues. And then finally dig deeper into the modeling of QQ, which is the glutamine, as a model for oxidized CaMKII. All right, so are there any questions? I know I went like 3 minutes over time, so I'm sorry.
Any questions for Daniela? I'll give it like a couple of beats. All right, if not, fantastic job. It was very clear. No one had any questions. So.
All right.
I think we had a chat question.
Everyone.
Oh, wait, was that a question?
Yeah, I think we have one in the chat.
Oh, there's a chat question.
Oh, okay, I see. Okay, how does CaMKII's molecular switch-like behavior contribute to memory and learning? So that's actually a really interesting question. And the lab that I work with focuses on heart implications of CaMKII. So I'm not an expert on how memory and learning in the brain is affected by CaMKII, but we're thinking it contributes to neuroplasticity. So this switch-like behavior is very similar to the, for example, all or nothing mechanism for action potentials in neurons. So we know neural activity works very much in the switch-like process, either on or off, either synapsing or not synapsing. So CaMKII kind of contributes to that mechanism by acting like another one of those switches in neural cells. So that's my guess, but again, not an expert on the brain aspect of CaMKII. But thank you so much for your question.
All right. This is officially the last call. I'll give it a couple of beats. All right. If not, thank you so much, Daniela, for your presentation. It was wonderful.